Okay, so we're going to finish glycolysis. Just this, which section is this? I forgot, 9.3. We're just going to finish 9.3. So the last thing we were talking about, we covered just the very beginning of glycolysis and talked about our 3 irreversible reactions and that they were very largely negative delta Gs. And we talked about our first step, and that first step was irreversible and required an ATP investment. So for 1 molecule of glucose, we had to put in 1 molecule of ATP in order to phosphorylate that glucose to make it glucose 6-phosphate. So we kind of talked about there being 2 different enzymes that could phosphorylate the glucose. In most of our cells, it's hexokinase. And if it's hexokinase, then the product of this reaction is an inhibitor of the hexokinase. But if we're not, if we're in the liver or if we're in the pancreas, we have glucokinase. And glucokinase is not inhibited by glucose 6-phosphate. And the question is why, right? So your body uses this enzyme, glucokinase, as an actual glucose monitoring system. So we talked about this system here. This is when you monitor your glucose if you're diabetic, right? But your body has its own glucose monitoring system. You need to know when you've eaten a meal, right? And so the way that your body tells the difference is to have 2 different enzymes. So if we look over here at our binding curve, right? We're looking at the comparison between glucokinase. So, right, here's glucokinase. And then we're comparing that to hexokinase. So if you look at this, there's a really big difference in binding affinities for glucokinase versus hexokinase for glucose. So if you look at hexokinase, hexokinase binds glucose at a very, very, very high binding affinity, which means that it has a very low Km value, which is what we see here. So it has a Km of 0.1 millimolar. So a little bit of glucose gets into your cells are basically going to phosphorylate it and keep that glucose to make ATP, or depending on if your ATP levels are high, you have other options. But when we're in the liver and the pancreas, we need something a little bit different because the liver and pancreas do something really, really important for us. They do different things. So first, we're going to talk about the differences, all the differences between the 2 enzymes. Then we're going to talk about why they're important. So we just said affinity, right? We just said that we have a very high affinity of hexokinase for glucose, whereas we have a very low affinity of glucokinase for glucose. So the low affinity, we're talking about like 10 millimolar, but that's still 2 orders of magnitude, right? So we also talked about this briefly, that hexokinase phosphorylates many different hexose sugars, right? It doesn't have to be glucose, could be other sugars. Glucokinase is glucose only. It's the only thing that it will work on because we want the body to have a glucose specific sensor. The other thing that we talked about was that hexokinase is inhibited by its product, glucose 6-phosphate, whereas glucokinase is not. And then hexokinase is in all of our cells, and glucokinase is only in the liver and the pancreas. So let's talk about why this is really important. Let's just say you haven't eaten in a while, right? So your blood glucose levels are basically right here. So let's see, right here. These are your normal blood glucose levels, about 5 millimolar. If you eat a meal that has enough glucose in it, enough sugar in it, to really spike your blood sugar, and that's usually what we do, right? Our blood glucose levels are going to go up. So our glucokinase inside of our liver cells and our pancreas are going to phosphorylate the glucose. And remember, the product, glucose 6-phosphate, will not stop the glucokinase from continuing, from keeping going. So why? Why is this good? Well, think about function. Liver. The liver's job is to trap glucose as glycogen. But do we always want the liver to be trapping glucose as glycogen? If we have just a little bit of glucose in our bloodstream, do we want to trap that in our liver? No, we want our brain to get that glucose. So we only want it to happen when we have really, 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 really high levels of glucose. And the same is true about our pancreas. We don't want to secrete insulin when we have normal blood sugar levels, because it's just not going to work for metabolism in our cells. We only want to stimulate insulin release if our blood sugar, again, is really, really high. And we'll talk in the next section about how insulin gets released when we have high blood sugar. Okay, but it's really important to know the difference between the two enzymes and how they function and all that kind of stuff. So our next reaction, reaction 2, is phosphoglucoisomerase. So in this reaction, all we're really doing is converting our glucose 6-phosphate, which is an aldose, right? Remember we said if it's an aldehyde, C1, right, is our anomeric carbon. We're converting that aldose to a ketose. Here, this one is C1, this is C2, whereas over here, this one was C1 because there's no other carbon attached to it. So we're going to go through, it's a multi-step pathway. And if you want to know the mechanism, you can read the book. The book has the mechanism for all of these reactions, but we're not going to cover that. But we're converting this aldose to a ketose. And if you kind of remember that basic pathway, I hope you have your glycolysis sheet out in front of you and you're adding this detail to that sheet. But you know it splits about halfway through. So in order to get basically equal things on either side of that split, we have to make basically a symmetric molecule. And that's what we're kind of trying to get to at this point. So we started in this reaction with glucose 6-phosphate. Now we're converting to a ketose, and that ketose is fructose. So we're going from glucose 6-phosphate to fructose 6-phosphate, right? And remember, this is one of those reversible reactions. And the reason it's reversible is that delta G is so close to 0. Now, our third reaction, very, very, very important. This is our major control point in the pathway. This is where we're going to do the most regulation of glycolysis in the entire pathway. So it is irreversible. Why is it irreversible? It's all about our delta G. Our delta G is very negative. So it's also our second point of ATP investment. The enzyme that catalyzes reaction 3 is phosphofructokinase 1. And we abbreviate that guy, thankfully, PFK1. So what are you actually doing in this reaction? Well, just look at your reactants. We're starting with fructose 6-phosphate. We're going to end up with fructose 1,6-bisphosphate. So all we're doing is adding an additional phosphate group. And look at your product. You're looking at something that's very, very symmetrical, right? If we draw a line down the middle, we're getting something that's very symmetrical. So this requires that input of energy. So we have to hydrolyze ATP to get that additional phosphate group added on to our sugar. So if we said this is a highly regulated step in the pathway, how is it regulated? Well, think about, when would you want to go through glycolysis? I want to go through glycolysis in my body when I don't have a lot of ATP, when my energy charge is very low. So this is low energy charge. So those are going to be positive effectors of PFK1. They are going to stimulate the activity of PFK1 to make more fructose 1,6-bisphosphate. If we look on the other hand at our cells, if you have a lot of ATP in the cells, we don't really want to go through glycolysis. And so a high level of ATP is going to indicate a high energy charge and is therefore going to be an inhibitor. They're all allosteric inhibitor, all allosteric regulatory molecules. So ATP, ADP, AMP, they all bind in the same place on the PFK1 enzyme, but they have different effects, right? So ATP is the negative regulator, ADP and AMP are the positive regulators. So super, super important step in the pathway. Aldose. This is reaction 4, and this is our cleavage reaction. So we have that name, right? Glycolysis. This is where the lysis in glycolysis comes from. So remember we said we kind of have that symmetric molecule. And so what we need to do now is we need to go in and actually cleave our sugar. And so we cleave it between carbon 3 and carbon 4. So I've numbered the carbons for us. And then we also cleave it between the anomeric carbon and the oxygen that's in the sugar ring. And so when you do this cleavage, you get something that's very close to being two of the same things, but not quite. So it's actually a 4-step reaction that uses a Schiff base as an intermediate, and you can see the whole pathway in the book, but we're not going to cover it in that much detail. So our two products are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This glyceraldehyde 3-phosphate, this guy, he is ready to go on in glycolysis. He is the right molecule. But the dihydroxyacetone phosphate, this one, the DHAP, this one is not in the correct structural format to be a substrate for the next reaction. So basically, our glyceraldehyde 3-phosphate, we're in reaction 4, is going to jump to step 6. It's done. So you can actually see that in your side-by-side pathway where one of those molecules of G3P, glyceraldehyde 3-phosphate, skips down to step 6. Whereas the dihydroxyacetone phosphate can't go to step 6 without going to a new step, and that step is number 5. Okay. So in step 5, we have our triose phosphate isomerase. So this one is just to convert the dihydroxyacetone phosphate into a substrate for reaction 6. And the substrate for reaction 6 we know is glyceraldehyde 3-phosphate. So it's simply an isomerization reaction. And we need glyceraldehyde 3-phosphate for the next step in glycolysis. So here's the end of ATP investment. We don't have to invest anymore. So if this is the end of stage 1, what have we done? We've really invested 2 ATP per 1 molecule of glucose. And what do we get out of this investment phase? We go from 1 molecule of glucose to 2 molecules of glyceraldehyde 3-phosphate. All right, so now we're in stage 2, and you could, add this stuff to your pathway. Stage 2 is all about making money, right? We invested money, now we got to make money. We invested 2 ATP. So in order to get a net 0, we need 2 to be paid out. And then in this pathway, we make another 2. So we make a total, of 4 ATP. And at the same time, we're going to make 2 electron carrying molecules of NADH, and we'll talk about those. But we're going to make 4 ATP per 2 molecules of glyceraldehyde 3-phosphate, or you could say you're going to make 4 per glucose, right? Either way is true. All right, so our very first reaction in step 2, in phase 2, is reaction 6 with glyceraldehyde 3-phosphate dehydrogenase. At least the name of the substrate is in the enzyme. So now we call this one GAPDH. There's so many abbreviations, you got to get comfortable with them. So in this one, we actually have a phosphoryl group transfer. So we're going to add a phosphate to glyceraldehyde 3-phosphate. So in this reaction, we're actually coupling 2 different things together. Number 1 is that we're going to reduce this NAD plus right here into NADH, and we're going to release a hydrogen. So we need these energy carrying molecules for other processes that we'll talk about later. But this is our payout for NADH, not a payout for ATP, but this is a payout for NADH. And we're going to couple that to the oxidation of glyceraldehyde 3-phosphate to make 1,3-bis phosphoglycerate. So if you hear the bis, right, what does bis tell you? It tells you 2. So you have 2 phosphate groups. You have one of them attached to carbon 1. You have one of them attached to carbon 3. So you have one here and you have one here. So that's where the bis phosphoglycerate comes from. But why is this really important? And this is the other thing to remember before I go on. I almost forgot to remind you. Remember, in the pathway, you have those 2 columns now in the pathway. So you really have 2 glyceraldehyde 3-phosphates, 2 1,3-bis phosphoglycerates, and you're actually making 2 NADH molecules there, right? So make sure you keep that in mind. And so if this reaction requires NAD+, right? This was NAD plus is something we had to put in to make this reaction happen. If you just go through glycolysis, you are going to deplete your NAD plus. And so you actually have to continuously replenish it, or glycolysis is going to stop. So you can either replenish the NAD plus through the electron transport chain, which we will talk about, or you could replenish it. And that electron transport chain, remember, is going to happen in the mitochondria. Or in the cytosol, you could use LDH. We all know what LDH is, lactate dehydrogenase. And we can use that to regenerate that NAD plus that's required in the reaction. So in this reaction 6, we're actually going to have energy being released from our oxidation of our glyceraldehyde 3-phosphate. And that's what drives the addition of the phosphoryl group to make that 1,3-bis phosphoglycerate. So this is really important, because what we're trying to do for the rest of the pathway is to make ATP. Well, in order to make ATP, we have to have substrates that have phosphoryl groups that are really, really high energy. And that's what we have in 1,3-bis phosphoglycerate. We have phosphoryl groups, and those phosphoryl groups have really high transfer potential. That's really, really important in order to make ATP. So can you guess what's going to happen in the next step? In reaction 7, we are going to make ATP. So the enzyme that makes the first real payout is phosphoglycerate kinase. So phosphoglycerate kinase, and remember, this same thing is we started with 2 molecules of 1,3-bis phosphoglycerate. Here, we're going to make 2 molecules of 3-phosphoglycerate, which means we're losing a phosphate. Well, where do we lose that phosphate to? We lose it to ATP. So we need 2 ADP, and we're going to make 2 ATP. So anytime we make ATP, what we do is we call that a substrate level phosphorylation event. Just means make ATP. So if you ever see that, you can replace those words with make ATP. And remember, you're going to make 2 molecules of ATP per molecule of glucose. So if we stop the pathway right here, we are at a net ATP investment, payout, whatever you want to say, of 0. We've put in 2, we've gotten out 2, okay? So this is a good point where we remember we're even. Now, there's one special thing that happens in red blood cells that's different than the regular glycolysis pathway. And that is that in red blood cells, our 1,3-bis phosphoglycerate is actually going to be converted into 2,3-BPG, right? 2,3-BPG. Remember 2,3-BPG bound to hemoglobin and inhibited oxygen uptake, right? So it was really, really important in making sure that our oxygen gets dropped off at our red blood cells when it should be. And so what's really interesting is that if you have patients who have defects in glycolysis, a lot of times they also have problems with oxygen transport in their red blood cells. And you wouldn't think that the two would be involved, because one's glycolysis and one's oxygen transport, but these are common shared substrates in these pathways. And so you end up having these overlapping effects when you have diseases, which is really, really cool. Okay, next reaction. Phosphoglycerate mutase. Phosphoglycerate mutase is just moving that phosphoryl group so that we have the ability to make another high energy substrate. So all you're doing is shifting around that phosphate group. Really simple, no big deal. All right, enolase. Enolase is our second to last reaction. We have a dehydration reaction, right? We're starting with 2-phosphoglycerate. 2-phosphoglycerate is something that is a phosphate ester, right? So this one contains a phosphoryl group right here, but it's just okay phosphoryl transfer potential. It's moderate. It's all right. But we want something that has a really high phosphoryl transfer potential so that we can have a very high negative G value and finish off our reaction and make sure that that reaction does not go in reverse. Because enolase, again, is another one of those reversible reactions. All the ones that don't have high negative delta G values are reversible. So our enolase is going to convert our phosphate ester into an enol phosphate, right? So remember enol, did I say enol? What an enol was before? I don't know. But you have the carbon-carbon double bonds. That's the ene, ene. And then the OL is for hydroxyl. So this is an enol, but it's a phosphate. Instead of having the hydroxyl, you have the phosphate. And this is a really pretty unstable molecule. And so it has very high phosphoryl transfer potential, which is exactly what we want for our last reaction in glycolysis, because we want this to be irreversible. We need the last step in the process to be irreversible so that we ensure glycolysis ends. Otherwise, it can go back and forth, and that futile cycling will happen. So our last step, pyruvate kinase, is irreversible. It also produces ATP, and we end with pyruvate, right? That's our substrate. So we know phosphoenol pyruvate is very unstable, but if we remove that phosphate group, and remember this is 2 molecules, so we need 2 ATP, and we're going to need 2 ADP, we're going to make 2 ATP, and therefore we're going to end with 2 pyruvates. Pyruvate's very stable and can be used for lots of different things in the cell. So this is a really good last step. It's going to decrease the energy of our molecule, but at the same time make us some ATP. So this is the end of stage 2. We invested 2 ATP in stage 1. We paid out 4 ATP per glucose in stage 2. We also paid out 2 NADHs in stage 2. And so our net is 2 ATP and 2 NADH in terms of energy carrying molecules, right? What did we also make? We also made 2 pyruvate, right? And we're not talking about other waste products. These are just kind of like big things that we want to keep track of. You can go and look in the very beginning slides if you want the whole chemical equation, but these are kind of the really important parts.